Oh, cheers. Good morning, living environment fans from all across the globe. It's Tuesday. What a great long weekend we had. I hope you enjoyed the weekend a little bit anyway. And yeah, cheers. Good coffee this morning. Yeah, we're in the week staring down the barrel of the midterm exam, which is coming up on Thursday and Friday, right? It's a 3-period exam. And yeah, hope you've been studying for that exam. And yeah, in the meantime, we're wrapping up cells. We're wrapping up our cells and microscopes lab and those kinds of things. So let's talk a little bit more about cells. Great coffee. Da 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 do 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 do. Da 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 do do. All right. Da 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 do do do. Hey now. Da 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 do do. Mr. Kovacs class. It's really fun. It's Mr. Kovacs class. You might learn a ton. It's Mr. Kovacs class. It's interesting too. It's Mr. Kovacs class. And he's super cool. It's Mr. Kovacs class. Yes, cells, glorious cells. At their simplest, right? It's just a cytoplasm, cell membrane, and some nuclear material. They're bubbles floating around, but they're special bubbles because they're bubbles that can utilize energy to process materials and make proteins. Sorry about that, I just jumped in there. Right, and yeah, cells. And we got prokaryotic cells, right, which are the simplest of the cells, the prokaryotic cells. And then we've been studying eukaryotic cells and all the different parts of eukaryotic cells. And then we've studied microscopes. We know that microscopes help us see cells because cells are so small, right? They're teeny. We've studied the difference between plant and animal cells. Of course, these are plant-like cells because you can see the cell walls. In fact, the first guy to see cells, Robert Hooke, this is probably what he saw. Then we know plant cells also have chloroplasts, like in these Alodia plants. You can see the individual little plastids, chloroplasts, little green bodies floating around. You know that's where photosynthesis happens. Right, and so we got, and then we've studied animal-like cells like this amoeba. I've showed you amoebas before, and we also learned that we need to stain them sometimes to see things inside. Like here's the nucleus, and there's some other parts that you can see inside this amoeba. And then you did that with your own cells, including your own cheek cells, where you stain them with methylene blue in order to see things like the nucleus. Right? Now, we're limited with what we use a compound light microscope, but I've talked about electron microscopes, which can allow us to see way more detail. Electron microscopes, right, can magnify hundreds of thousands of times, and you get to see different things going on. And because of the electron microscope, we know a lot about the organelles, or the little baby organ parts, cell parts found inside of cells. On an electron microscope, the images are always going to be black and white. Do you remember why that is? Good, right? To be colorized, you have to go back and colorize later because electron microscopes use electrons instead of light. And you don't get color without light. All right, so here's chloroplasts in a light microscope. Here's a chloroplast in an electron microscope. You can see much more detail on the inside of the thylakoid membrane and all that stuff, right? Yeah, electron microscopes allow us to see a lot of detail. Here's a mitochondrion. Again, it's been colorized. It's not really green. And then actually outside the mitochondrion, we can see lots of endoplasmic reticulum with ribosomes lined up on it. We could see a couple of what look to be like lysosomes and some other cell bodies in there. So that's pretty cool. The Golgi apparatus, of course, like a post office, packages materials and moves them. Discovered by Alessandro Golgi. All right, Golgi apparatus. What else we got here? Here's some more ribosomes lined up on the endoplasmic reticulum. You got some vesicles, most likely lysosomes. You see them smooshing up against the cell membrane to release there. So these vesicles are most likely got enzymes in them or something the product of the cell made, and they're releasing them to the environment. All right, and then of course there's some other things we're going to touch on. We haven't touched on centrioles or the microtubules that hold up the cell, but we'll talk about those, especially when it's time for cells to divide. It's really kind of a complicated process, and you need to coordinate some of these cell parts, all right? So yeah, cells, glorious cells. Class, of course, we do the Bellringer. We have an Ed puzzle, and we're getting ready for the midterm this week, so we'll do a little review. And yeah, that'll get us through today. Should be a good day, glorious day. If you're in lab, we're going to finish up the cells and microscope lab today. Right, some of you need to finish that up today. If not, we'll finish it tomorrow in lab. Okay?